Hello, Tharun and Akshay.
Hi.
Hi, Bradley.
Hi, how are you?
I'm fine.
Good. Wait for
Is Susan joining? I have some doubts regarding the paper.
Oh, well, I don't think she's joining today. She said something about her computer being, she had to have it fixed, so I don't know. But I'm gonna, I can probably answer some of the questions too, so. I have some, I have a picture of what she was talking about last week, so. Yeah. Hello, Mainak and Aayush. Hello. You're muted, I think. I don't know, your audio isn't working somehow. I still can't hear you. Okay, welcome to the meeting, everyone. We'll probably have a couple more people come in. We always do. There's someone right now. That's Akshay. So today we're going to talk about follow up on some things from previous meetings. We'll have a couple people said they could present something. I don't know. I think Tharun wants to present something. And,
I'll be presenting.
Yeah. And Mainak maybe has an update on some things. Is that correct?
Yeah, yeah.
Okay. So,
and can you hear my voice now?
What was that?
Can you hear my voice now?
Yes.
Actually, okay.
Yeah. Okay. So, first though, why don't we, and then we'll have some other things. I have some presentations and some papers, and we'll go over our deadlines. If you're watching on YouTube, you'll be able to follow or catch up as we go along here. Hello, Mayukh. So why don't we have some of the new people introduce themselves first? I think we have, I don't know, I think Tharun introduced himself a couple weeks ago. And Akshay and Aayush, I don't know if they've introduced themselves yet, so. Oh, your audio's really low. It's all the way off now. If you want to, you can type in something in the chat too. Why don't we have why don't we have Akshay introduce himself first?
I actually gave an introduction last week.
Oh, okay.
But if I think Aayush is new, so I would just introduce myself. So actually, I am Akshay Nair. I hail from Kerala in India. I'm currently undergoing my undergraduate studies in computer science and engineering. I am deeply interested in working with open source organizations, and the previous organization I've been working with was KDE. It's actually an application oriented software, not totally biology related. So yeah, this is a new challenge for me. Yeah, that's it.
Well, good. Well, thank you for going over that again. And Mainak says hello, Aayush.
Yeah, am I audible now?
Yes, you're audible now. Okay, good.
Okay. There was some problem with my headphone, I guess.
Oh, yeah.
Okay. Okay. Then I start my introduction. Hello everyone. My name is Aayush Kumar, and I am a final year undergrad at KIT University, Bhubaneswar, India. And my research interest mainly lies at the intersection of deep learning and AI for healthcare. And previously, I have worked at IIT Bombay and IIT Kharagpur during my second and third years. And last summers, I started working as a visiting undergraduate with research intern at Harvard, where basically I worked on connectomics data, that is basically the mitochondria and small cell organelles and mitochondrial segmentation. And then after that, I worked on the volumetric segmentation of adult Drosophila muscles. So basically what we did was we developed a deep learning segmentation model, a 3D model, and for carrying out volumetric segmentation, and after that we incorporated that deep learning model into a fully and Fiji pipeline. Fiji is basically a Java-based software which is used by biologists and microscopists to view the data, volumetric data. So basically, I worked on that, and once the 3D model was developed, we delivered that product to NCBS. It's basically a National Council of Biological Sciences, which is based in Bangalore. And since then, that work is over. I'm currently working to improve the segmentation task further by using coordinate convolutions as well as anti-adaptive aliasing methods.
That sounds good. Yes, very good. Ujjwal, hello? Ujjwal's here. So why don't we start with the presentation? So Tharun, if you want to go first, maybe, and then we'll go to Mainak. I think Mainak is still there.
Just a small presentation.
Okay.
Can you see my screen?
Yes.
Yeah. So I'll be talking about diatom tracking using object detection. So the main motivation here is to study the non-neural cognition of diatoms, that is given an input such as the intensity of light, the concentration of CO2 and the nutrient concentration in substrate. We want to study the responses of these diatoms. As movement is the most apparent response, so it will be very much easier to study these. So we need to track the objects across multiple frames, finding their relative position with respect to other diatoms and their velocity. So one approach could be using OpenCV methods such as thresholding and masking to find the position, but these won't generalize well across as the background might change, the light illumination and the resolution of images. So as we know that neural networks generalize well across multiple domains, so we'll be using neural network based object detection. So the most naive approach to detect objects will be to use a sliding window where we just take a part of an image and run CNN classifiers through it to classify whether there's an image or not, and another neural network to predict the coordinates and the width and height. But the major drawback will be that we need to do multiple passes over the image to get all the objects, and when we pass a part of the image through the CNN, it is possible that the whole object is not present, as you can see in this right image. So one proposed approach was to use a YOLO based object learning model, which treats the classification model as a regression model. So basically it divides the images into a grid. So where here you can see it's a 3x3 grid, and assigns each grid to an object whose center lies inside that grid. So each grid makes a prediction of the probability of an object being inside the grid, the coordinates at the center of the object, the width and height, and the class label. So for example, in this grid, if you're trying to find whether there is a car or not, so there is not, so the probability will be 0, and the other predictions we won't care about. But in this cell, as you can see, there is a car, but this is placed in both these two grids, but the center of the car lies in this rightmost grid. So this grid will be making the predictions of the center of the object, the width and height, and the class. So the YOLO object, by dividing the image into several grids, makes it possible to find almost all the objects through just a single pass. So we get a fixed number of bounding boxes prediction from a grid. So we don't need to make several passes through the image, so it makes it much faster. So this will be one of the main reasons that we choose YOLO for object detection, because it is fast, so it can make real-time predictions, like 45 frames per second, or like 145 frames per second depending on the model. And YOLO is trained end to end, so the parameters are shared between the classifier and the bounding box predictor. So it has much more higher accuracy than those models which are having different models which predict on the classification and bounding boxes. So these are some of the predictions that the model which we trained on Diatom object detection. And certain drawbacks that we can observe in these images are it fails to detect the objects which are very much close to each other because of the grids which can only detect a certain number of bounding boxes which are fixed. And so sometimes it might be possible that 2 or 3 atoms get predicted together into one single bounding box, as you can see in this right bottommost image. And the pre-processing part also for preparing the data set becomes much more difficult, as these object detection models require the bounding boxes to have their sides parallel to x and y axis. So you have to rotate the object in such a way that the bounding boxes which we predict are always parallel to x and y axis, and this becomes much more time consuming, and we cannot automate this, as the image needs to be manually rotated. And when if you have a bunch of diatoms which form a structure and they are bent, so it becomes highly impossible that we can rotate the image in such a way that the bounding boxes are parallel, or the bounding boxes are parallel to x and y. So we cannot detect all the objects in a single pass. So what we can do, we are further planning to do, is to modify the algorithm in such a way that we can increase the number of meshes, so that multiple bounding boxes can be predicted, and also to increase the data set size so that it can generalize much better across multiple images. And to overcome the pre-processing part of the images, we could rotate the images in all possible angles and predict the bounding boxes and stitch those predictions together to get a single bounding box per object. But this algorithm will become much more complex and become computationally expensive, as we are rotating the objects in all possible images in all possible angles and then making predictions. And one more model which we can try is using instance segmentation, where the model makes pixel-wise predictions of, so it predicts the class of each pixel. But this preparing the data set for this becomes much more difficult and time-consuming. We have to draw the outline of each object, which becomes difficult when you're using a mouse or trackpad, so we require some special equipments. Yeah, so that's it. So any questions or suggestions of how to improve the model are highly welcome.
Yeah, that was
Okay,
very
so-
good. Oh, go ahead.
Thank you, Tharun, for this presentation. I actually had a question. Could you just go back to the slides, please?
Yeah, one sec.
Okay, sure. So could you go back to the slide which was talking about the bounding boxes? I mean the portion where you were talking about the the rating part. Yeah, this slide. So you have discussed about this thing where you're trying to rotate and make every picture basically vertical with respect to those cells, right?
Yeah,
Because yeah, I get it. So and if you rotate it, then the bounding box does not actually cover the thing itself. Am I right?
sorry,
It becomes a diagonal sort of a thing then. So
no,
I think what you can do instead, like you talked about this, like I think what I had an alternative in mind is that like you can use a segmentation map and then like use random rotation. Like in this case, you cannot really augment the data set other than vertical flipping, right? Or some transform some translations like shifting in X and Y plane. So instead, if you want to go really heavy on the augmentation, you should go for segmentation models.
Yeah,
see, you're telling me that like segmenting it on a manually annotated data set is really like not good. But what happens is that like we can just experiment on it at first and see how it performs on a test set.
No, like preparing the data set was a problem. Like to create the boundaries of the objects, like we couldn't do it. I tried doing it with using a mouse and trackpad, and like it was
So
not that
perfectly accurate.
Yeah.
Right.
That was like
so.
Yeah.
So what we can do instead is that you can use these bounding boxes maybe to see. Can you register those rotations? Like you're rotating all of those to become vertical, right?
Yeah.
You're rotating it using Python, so you can reverse the rotations, right?
Yeah, we can reverse. Like in by predicting, no. If you have like we predict on an image or a video, then we have to rotate each individual frame, no.
Yeah, exactly.
To make it.
You can rotate it. That is fine. But the thing is, if you can rotate it, you can also rotate it back, right?
Yeah. So
So to generate the segmentation data, what we can do is we rotate it vertically, as you said. We generate the bounding boxes with your model, and then we can use that bounding box to create the segmentation mask by filling that bounding box. And we can rotate it back, right?
Yeah. We can do that.
Then we can rotate it back, and then we can do all sorts of augmentation. I think that will solve your manual thing problem.
So you're like telling us to create a bounding box, but use the instance segmentation model by rotating back.
No, for annotating data, we can create the bounding box, and then we can fill that bounding box, and then we can rotate it back to whatever it was, and then save that data set like with pairs of images and segmentation masks, and then augment and do whatever with it for training.
Yeah, that would be for instance segmentation.
Yeah, instance segmentation. That's what we're doing, right?
Yeah.
Because see, it's more segmentation is much more useful than bounding boxes in general.
Hello, am I audible?
Hello?
Hello, am I audible?
Yeah, you're here.
Yeah, so basically like there was a problem in my mind. So basically Tharun Mayukh is trying to say is we have explored the YOLO V3 like bounding boxes approaches, but ultimately like we have to go for the semantic segmentation. Instance segmentation is also not going to work for us. We have to go for semantic segmentation. And the thing manually annotating the data set. So basically if you are making.
Oh, you dropped out, Ujjwal. Hello? Can people hear me? Hello?
You can see bounding boxing approaches is not going to is generating result, but we have to select particular images. This was the same problem that we have faced 3 years ago when me and my friend are working on this project, and we have also rotated all those images and inverted it back, and we have done semantic segmentation and that kind of thing. So if you are going with bounding boxes and you are trying to rotate it, the data, my understanding says when you have created the bounding box by rotating the image, then you re-rotate it to its original form, and whatever bounding box you have get, you can just mask them, make a binary image of that, so that you can get the annotation for that each cell. But that is not also going to be work in the semantic segmentation case, because this is not it is still a good result, but as you can see in the elongated cases and different cases, it is not giving us very good results. So what you have to do is it is a tedious task, I know, but you have to label images using different labeling tools. I know in those labeling tools you still have to rotate the image, because they also support only X and Y axis, and they don't support the cross access. So that is the one way. So for anyone, like if anyone is going to work for this project, like Accelerator project, so like please kind of be prepared. Like you have to annotate data, like because without annotating data, you can like create all these kind of binary masks and these kind of things, but you still have to like create good annotated data because that was a part of the project. Because without that, semantic segmentation, like if you do bad annotation in the first step itself, the further model is not going to work very well. And that I can say from the experience that I have done in this project. So like you have to annotate images very good. Once you have very good like there is not required like a very huge annotation. Like I think 200 300 images can work, which can be done in a week or so. And after once you have get those images, then you can work with the semantic segmentation, and it is going to produce very good results. And even with this bounding box approaches, what I'm trying to say is you can still like all those images that you are showing, like where the diatom is very much elongated, you can ignore these images. Like we have, like the frame rate that we have taken is 1 frame per second, I guess. What I remember from that time, you can take very frequent frame rate as well, which will help you create a very large data set, and you can ignore all these images. You still can get these kind of images where you can perform. Yeah, this kind of images where these are performing very well. These are very actually pretty good results. Like when we have worked on with Deep Lab, we are not getting these kind of good results. And I think many things have been improved in 2-3 years. So we can also do that kind of thing. So that was my feedback. Like I think right now you are on right track.
Yeah. This set was like only to zoomed images, so it performed very well. It like you almost
Yes,
like
yes.
85% MHP score, but it failed on this part.
Elongated set, yeah. And if you when we are going to like give you data and all, you will see like these elongated fields are very less in amount. These images with zoomed out images are very much in the video, and when we are extracting frames, I think we can get what we are trying to do.
Yeah.
Yeah, that sounds good. Thank you, Ujjwal and Mainak and Tharun for presenting. Yeah, I didn't really have any comments. I mean, it looks good. I was gonna maybe raise some of the comments that other people were raising, but I think we got that taken care of. So we got some people. Yash is a new person in the Slack. He's here. Welcome. Assaf is here as well, and Shruti is here. So now it's time for Mainak to present the thing he wanted to show. Can you share your screen?
Yeah, absolutely. And can you hear my voice?
Yes.
Is it okay? I'll share my screen real quick. Okay, can you see my screen?
Yes.
Okay, great. So if something happens, like if the screen goes off in the middle, can someone let me know? Because my network is having some issues. Is that alright?
Yes, it looks good.
Okay, so I'll begin with the presentation. So this presentation is basically an extension of my conversation with Bradley and Mayukh. So I have been working with UCAT cells a lot lately. So the thing is that I've been trying to reconstruct the continuous processes that are undergone by these cells. So I'll be exploring these avenues now. So I'll move on with the next slide now. Okay. So the contents of this presentation, yeah. So first I'll be speaking a bit about the UCAT cell cycle. Then I'll be speaking about the deep learning module itself and the aim of it. Then I'll be speaking a bit about the training process itself and the details of it. And then I'll be speaking about testing the module and extracting inferences from the training module. So moving on to the next slide. Okay. So this is the UCAT cell, as you can see, like in the bottom of this figure. So this is basically an immortalized line of human T lymphocyte cells, and these cells are basically used to study acute T cell leukemia and T cell signaling. So these cells, they undergo a cell cycle which has 3 stages. the first stage is G1 stage, the second stage is S stage, and the third stage is G2 stage. So these processes, right, these cycle, so this cell cycle, it's actually a very gradual process, but the labels that we got in the data, they are just labels. Like they see, the labels that we got, they actually do not capture the gradual nature of this cell cycle process. So we'll be exploring that soon. So I'll move on to the next slide. Okay, so the network that I have, it will be trained to basically classify the stage of the cell cycle from the cell images. So as you can see here, here are the three cells. The first is the G1 stage, the second is the S stage, and the third we get the G2 stage. So by looking at them, we don't really, we can't really figure out which stage they are in actually, but the trained neural network can actually. So I'll move on to it. Okay, so coming to the pre-processing part. So this part basically ensures that the data is suitable for the PyTorch data loaders. So the first flaw that I found in the data is that the data was imbalanced. So the thing is that the data that I had in its raw form, it had a 45.4%, like the class G1, it had a weightage of 45.4%, which is very high compared to the others. So what I did is that I basically oversampled the minority classes. So I basically oversampled the minority classes to get even number of samples from each class. So okay, so I also divided the dataset into 10 folds. So I'll be training. See, my plan was to train 10 different models on each fold. So the training would be something like this, that out of these 10 folds, in the 9 of the folds, the first model will train, and in the remaining fold, it will validate its performance. And for the rest of the models, it will be something very similar, but with a different set of folds. So yeah, and I also left out 2503 data points as a holdout set for future testing. So moving on. Okay, so these are the training images that I had. They were the actual images were 66 by 66. They were actually RGB images. But I center cropped each image to 32 by 32 to get rid of unnecessary background pixels. So, and I have also used heavy augmentation techniques using a library called albumentations. So I guess that will help in the overall generalization of the model. So I have also used Optuna to automatically find the best training hyperparameters. And speaking of the network itself, I opted for a ResNet 50 backbone with custom linear layers at the end, because the ResNet 50, it has a lot more classes in its layers, in its linear layers, but here we have only 3. So I had to make those changes, and I actually trained 10 instances of the model on 10 different folds of data, as I had said before. So moving on. Okay, so after training, I used the 2503 holdout points for training, not for training, to actually benchmark the performance of the models. So while running inference on only one model, we got an accuracy of 85.5%, and when I actually blended the inferences from the 10 models, I got an accuracy of 88.09%, which is actually slightly more than the one before. So as it turns out, when I blend the inferences, I get a bit more accuracy, which is good. So here are a few samples with their corresponding labels and the predictions. So these are correct, of course. So I'll move on to the next slide. Okay. So dimensionality reduction using Umap. So this part is a bit complicated. It took me a while to wrap my head around it. Anyways, I'll attempt to explain it to you all. So each image is basically a vector which has the dimensions of 3 by 32 by 32. It's a very high dimensional vector, right? So these vectors can be reduced into only 2 dimensions. So in this plot to the right, every point actually represents one single point in the test data set that we had, which consisted of 2503 data points. So each point here represents an image from the test data set, and the color represents the predicted class. So the special thing about it is that, okay, I'll test that on the next slide. So yeah, okay. So these images, right? They were classified, and these colors represent the classes they were classified into. So as we can see that these dots, they merge into one another, right? Like these vectors, these images, these classes actually merge into one another. There's no solid or there's no abrupt line between the classes. They are like it kind of represents the gradual nature of the process that we were talking about before. And there's also considerable variation in this blue segment, which shows that the G1 class images, they have a lot of variations in them. So this broad spread actually represents that. So another special thing is that at no point while training, we specified the order of the processes, right? We just told the model that here's the image and here's its label. We never said that these labels, like, we never told it the stages and the sequence in which it undergoes these stages, right? So like the model itself came up with the conclusion that this S phase comes in the middle of the G1 and the G2 phase. So that itself is pretty spectacular actually. So like it could be also like functionalities like this can also be expanded into other fields like some other cells or some other organisms where we got similar image data sets. So like it's actually really interesting to have these reconstruction of processes from trained neural networks and having the ability to know the stages in a process just by training a model. So that's it, I guess. So that's all from my side, and I'd love to hear some feedback or questions that you guys might have now.
Thank you very much. That was good. I had a well. Actually, could you bring the slides back up? Just reshare your screen. Yeah. As I wanted to look at, make maybe some points about it. So, I like the Umap analysis. He showed me that in the Slack a couple days ago. So this is the Umap, and of course we talked about this in a prior meeting, like how Umap works. So I refer you back to that presentation, and they have a new version of this out now. So if you're interested in methods, that's a good place to go. But so this is basically a dimensionality reduction of your cells and their different phases and how they're sort of related to one another. So they overlap. So I mean, the basic logic here is that if they're separable groups, they'll be like clusters, and if they're not separable necessarily, they'll blend together. And this is sort of because this process is, it's the same cell. It's just going through these stages, and you can identify it using the phenotype. Then you get this blending across this arc that you see here. I don't know if there's any significance to this arc, but it's definitely showing.
No, it's actually really it's not really significant about this arc. What we can infer from this as a rule of thumb is that if there are two different points, these two different points actually refer to two different images. So that's the basic thing. But this arc that really has no significance here actually. The shape really doesn't have significance.
Yeah. But this is basically the important part is that they blend together. So I mean, when you look at any one image of cells, unless you synchronize the cells in the culture or wherever you're looking at them, they usually have like cells in many different stages across the image. So like one image would be like one snapshot in time of a cell culture, and those cells, unless you synchronize them using some sort of like technique that people use, then you get like cells that are in different phases or in different transitional points. And so, you're going to get like, it's not going to be like you can have everything in G1, everything in G2. They're going to be, So this is good. Yeah.
Yeah, so what I wanted to add to that is that, see the labels that we had, like the labeling procedure that they followed, they actually, they have basically assumed that like under each label, every image is homogeneous. But after training the model itself comes to the conclusion, like from this Umap from the stream map plot and from the prediction colors, we can completely infer that the images that we have, they are not homogeneous if they come under one class label, right?
Right.
So that itself is pretty interesting actually. Like, we usually assume that under one class label the images have to be completely homogeneous, but this plot shows that by using these approaches we can infer the gradual nature of these processes. That's all.
Yeah, and it's it depends. Now I don't know. You didn't. One thing you didn't do is tell people what Jurkat cells were. I don't know if you know what the. I mean, sometimes they give names to cell lines that are like kind of. I can't remember. I looked it up before, and some sort of model. We have model organisms. We also have model cell lines. So it's some sort of cell line that's. Oh, there we go. We have this. So this is the. Bring that up. Yeah, a mortalized line of human T lymphocyte cells that are used to study acute T cell leukemia, T cell signaling. Yeah, so this is a model cell type. It's something easy to culture, and so this is so these cells are going to be, you can, different cells have different growth rates, different phenotypes, and so, I think it's good that we have like these model cell lines because we kind of know how they behave, and actually for like if you're doing something like this, it's good because you can, maybe build a model where you can characterize what the cell looks like, what things should look like in the cell. See, like here you have the G2, the S, and the G1 class, and they all have like very distinct phenotypes. Some cells don't necessarily have that. So I think that was a good choice for that.
Yeah.
Did we have any other questions about Mayukh or anyone else?
I didn't get it, like what you have done for balancing the unbalanced data, like what is like you have done?
Okay, I'll go to that slide. Okay, can you see the slide?
Yeah.
Yeah.
Yeah, so the data set that I had, it had a lot of images, right? So for the G1 class, I had, so for the G1 class, the 45% of the data was only the G1 class. And like 27% were for the S2, for the G2 and the S class. So it was actually heavily imbalanced, which had to be balanced to make sure that there's no inherent bias in the trained model. That's what I meant.
So like what are the steps? Like have you just picked up the random samples and discarded some data? Like what are the steps for that?
No, I did not discard some. I did not discard any data. I actually oversampled the minority classes. Suppose we had 100 samples for G1 and 70 for G2. So what I did is that I took 30 copies of G2 and just
Sure.
So basically, exactly. I just oversampled them. That's all.
So instead of doing oversampling, because oversampling is always going to add some sort of biasness in your when you are training, you can use something like stratified splitting. Have you heard of it?
Yeah, exactly. I've actually used that. I've actually used multi-stratified K4 from Katyal. Yeah, I've used that. That's great actually.
So because oversampling is going to induce some sort of biasness in the dataset, so I will prefer
especially in the folds, right?
Yeah.
We are speaking of the folds, right?
Yeah.
For creating the folds itself, I had used MKSF, which is multi-stratified K-fold, so that takes care of that bias part that you're speaking about. So it's taken care of.
Oh, good. Thanks.
Okay, so anyone else? Any questions or feedback?
Okay. I mean, if people have feedback for Mainak, we can talk about it on Slack. But yeah, that's excellent. Excellent presentation. And Tharun, excellent presentation. I know you guys have been working on this, and this is great stuff. So why don't we follow up on this in the coming weeks and see how it goes. I wanted to start going into some of the things I had prepared now. And there are a bunch of things. I know Susan isn't here today, and she had some computer problems, but she sent me some images, or an image of her ball microscope. Okay, so we actually have something in the chat here. I didn't look in the chat. Mayukh says the neural network learned the continuous representation order without the user explicitly feeding it into the model. Interesting. So yeah, this was where we did. It's sort of this sort of self training or yeah, self supervised type of thing going on here. So yeah, we'll follow up on this. So hi, hello Krishna.
Hi Bradley.
Hello.
Apologies for getting late. Actually, I had to, I was working for a conference, so I'm just here to say hi.
Okay. Yeah. Hi.
A lot of new people here, right?
Yes.
Hello everyone. Hello Yash. Hello Tharun. Hi Mayukh. Hi Ujjwal. Hi Mainak. And happy birthday, Shruti. It's her birthday today.
Oh, happy birthday, Shruti.
Yeah. So okay, so you guys continue. I just wanted to say hi. So I had a conference, so see you in the next week. So how many of you are for GSoC here? Hello?
I think a lot of people are here for GSoC.
So,
Probably, well, yeah, all the new people, I think.
okay. So I hope you people got the Google Doc and the slides for GSoC. Bradley, did you share them the onboarding files?
Actually, I'm going to talk about that in a minute. Yeah, we have some things to get. Yeah, we have some things to organize and send out. So I'll be sending those things out this week. So let me see if I can pull them up here. So we have. All right. So this is the onboarding. So right now we have 2 onboarding documents, and I'll put these make these more prominent in the Slack. The first is this thing that. Krishna put together, and this is on open source, sort of the open source motivations of our group and of Summer of Code. So this is a short PowerPoint presentation, and it kind of goes through what is open source, why should you contribute to open source, and then a little bit about Summer of Code, the deadlines here. What a bit about DevoWorm. Want to contribute to DevoLearn. So this is a set of ways in which you can contribute. Of course, you can just do your, you can come to the meetings, or you can do a project proposal where you can get selected and do a project. But there are other ways to contribute to DevoLearn and DevoWorm. And I'm going to go over these slides and edit them a little bit before we do them. We put them up. The other thing, of course, is the thing that Mayukh sent me, which is about specifically about the projects. So we have the project descriptions up on Neurostars and on GitHub, but we have actually a little bit of like, what can I do before GSoC? So GSoC is roughly this application period, which we have in the slide. So you have the deadlines there. But, there are things you can do before you write your proposal. So you're going to be writing a proposal for GSoC. What can you do before then? And so he has these things here listed, what you can do before GSoC starts for each project. That's very nice. And then the skills and requirements here that you might want to highlight in your proposal, or if this is something that you really specialize in, you might want to make sure everyone knows that, or get ramped up on it before GSoC. And so there, this is, I'm going to make these available here. There's also a general FAQ, that FAQ that he put together. So thank you for this, Mayukh. And we'll probably be putting these materials up now. I'll go through them and edit them a little bit, and then make them publicly available in the Slack channel. Okay, we have people coming into the document. Okay, so that's good. So yeah, and I think that should be good, I think, for onboarding. We'll have, the very specific open source part, the general part about the project. So I think that's good. And thank you to both of you for putting that together. And so I also wanted to point out here something Susan sent me, and this is for the third project here. This is the ball microscope. So if you can see my screen here, this is what this is a microscope that images like an embryo from all sides. And so what happens is you put your sample inside this ball. You open it up, or actually I think it's down here. Well, this is the light source, I think. But your sample will be loaded around here. And then all of these are different cameras here. I don't want to comment. These are different cameras here hooked up to a capture device. And they're basically taking images from different angles. So you can see that they're coming up from below, coming up from on top. The light source is coming in here and the sample's in here. And so it's able to image simultaneously from all these different angles. And so now that we have all these images from different perspectives, we can then take all those images and stitch them together into a sphere. So you can see that you can capture information from various angles on the upper hemisphere of the embryo, and then different angles of the lower hemisphere of the embryo. And then one of the goals of digital microsphere is to be able to take those data that are being collected and stitch them together. Now, I think I made the data so Susan has some data that's available, that she made available to me, and I put them in a secure server, and I think I sent the link to one person who was interested.
That's me.
Okay, so that was Akshay.
Regarding that, like, I have actually the doubt, like, I was actually thinking about montaging pictures, so now it got clear, like, you were talking about stitching, so now it's totally fine. So, the subsample you sent, like, is it possible to create at least a demo for just the proposal, just for me to visualize at least?
I think I might have some animated GIFs, but that's about as far as I've gotten on it. Just making a demo. But it's basically where what happens with this, like, each set of images is, so there's like, I think there should be like a folder of like about 12 or 18 images per embryo. And basically what happens is it's like you're taking a globe and you're spinning it along its axis.
Yeah.
So you'll see that those images are in sequence, and if you stitch them together, it's kind of like it's rotating. Now you can do things with that. You don't need to build like an animation just from those images. You can montage them or stitch them together in some way. I mean, the way that you might want to think about this is like a geometric projection, where you have, if you make a flat map on a surface, that flat map has been taken off of a globe, right, off of a sphere, or off of a curved surface, right? And you're deforming that surface, but you don't want to deform it too much, or else you won't be able to tell what the features are on the map. So you want to be able to, create like you can montage the different images. You want to take like the coordinate system and you want to deform it enough so that it flattens out, or you can sort of see it clearly, but you don't want to deform it too much. I mean, that's basically the idea. So in the proposal, I think we said that students can come up with their own solution to the problem, and then it would be evaluated. So I mean, you have a lot of freedom in terms of how you want to approach it.
Yeah. And one more thing, like the subsample you shared, is it over an instance or something, like what the research paper like written by Susan and Richard, like I went through it like completely, and they told that like the 3D images over time it becomes 40. Then we would be it's actually the fourth dimension is the time, right? So I was thinking whether you send me the images of an instance, or is it just a random subsample?
I think it's a random subsample. I think I gave you like one folder that she gave me. I'm going to have to go through it again and look and see exactly what's in there. But I think it's like it's just one instance. I would just, we can go through it later and see what's in there. But I think it's one instance. I don't think it's like a time series or anything like that.
Okay.
Yeah. And so if people want to, people want access to this secure server that we have it on, let me know and I can send you the link. I think that's a good way to distribute data. I don't want to make it public yet, so we'll just do it that way. So very good. Let's see, we have some more things in the chat here. Okay, Ujjwal asked, Akshay, are you using 3D rendering models or trying it in Blender or Photoshop?
Well, like I was thinking of using OpenCV actually to sketch images. But yeah, I would give it a look. Like so far, right now, I've got an idea about what the project should be. Like after going through the research papers, and you clarified some doubts I had. So I would be using OpenCV for it. Like right as of now. Like if it is not possible, then we would be moving to other alternatives. I would be going through it. I have a plan in mind, so I'm actually preparing my proposal also right now.
Yeah.
I hope Bradley gets time to review it. I'm not sure.
Yeah, I will review people's proposals when they have them. I can give you feedback on them. So just again, drop a link in the Slack on a private DM. It'd be fine. So yeah. So let's go on here. I have a lot of things I was wanting to cover. We'll probably get through them. I'm gonna very quickly.
Just one quick thing I wanted to ask.
Okay.
Hello.
Yeah, go ahead.
Yeah. Hi. So a few days back we were talking about the cellular automata thing, like I told you regarding different models that we can try. So
Right.
I was wondering that if you could, if you have some resources, you were telling me in the near future you might be presenting something or something like that. So lately I've been a little busy, so I could not do it a lot. So yeah, if there are some resources that you would have in mind or something, I would love to have a look at them and get some idea about it.
Yeah, we're going to talk about that in a couple minutes. So we're gonna.
Okay.
I'm gonna go over that.
That's great.
Yeah. So, but I wanted to go over a couple things here before that. The first, I'm not sharing my screen anymore. Okay. So the first is just briefly to touch on the submissions. So we have, again, some deadlines coming up. ComplNet, which is March 26th. The Biosystem Special Issue, the Periodicity, and the Embryo, that's been resubmitted already. Thank you, Ujjwal, for your comments. They've been incorporated, and Jesse as well. We had, I think it came out pretty well. We'll be waiting for that decision pretty soon. So I hope it gets accepted soon, but we'll see. This INCF Neuroinformatics Assembly is coming up in April, and the deadline for submissions for that is March 31st. And they want a 1,500 word or 1,500 character abstract. And so I've actually prepared something for that here. This is on the DevoLearn platform. So I think I mentioned this briefly last time. We're going to walk through this DevoLearn platform. This is the presentation that we would give. I would take the lead on it. And just kind of go over the platform, talk about the different components, and then finally discuss a little bit about future development in terms of these epistemological directories, which, would be something that we would go over a little bit beforehand in the group. I'll probably give a presentation on that. And then also further development of some of the software. So I'm going to submit this. I can send a link to this for people to edit or to look over before I submit it. But then we can submit it, and hopefully we'll be presenting in front of INCF. I mentioned in here that part of the DevoLearn sort of development has been sponsored by INCF. So hopefully they look at that and say, oh yeah, we need to include this in the symposium. So we'll be revisiting this. Also, I think Krishna had to rejoin, but I wanted to point out that his work that we've been doing on ANNs and BNNs, which are the artificial neural networks and the biological neural networks. We're doing a paper for the Artificial Life Conference on that, so we're going along on that. There's still a lot of stuff to do, but this is something that is due probably next weekend, so we'll be kind of I'll be cranking through that this week, and hopefully maybe people want to give feedback on it. I can send it out later this week when it's almost complete. People can read it over and look at it. And then I think that's it for the submissions. I think there isn't too much else coming up very soon. NeurIPS actually is coming up in May, the deadlines for papers. So if people want to submit something to NeurIPS, it's very competitive, but if you want to do that, that deadline's in May, so it's coming up soon. So just to keep that in mind. And then the evolution abstracts are now open again, and we can submit something there by April 30th. So now I want to move on to the cellular automata stuff. So this is Shruti's. She just asked me about this, so I'm gonna. I have some resources here that I want to point out, and this is a very crude sort of introduction to it, but I wanted to show you what we've been doing, what we've done in the past in the group here, and some of the stuff I've done in the past on it. So we have this paper, and this is available on the DevoWorm.weebly.com website. That's the group website officially, and this is in the Publications tab. It's Morphozoic Cellular Automata with Nested Neighborhoods. So this is Thomas Porteges, who I think gave a presentation in the group last year. He was a more active member several years ago, and he's come up with this technique for cellular automata called Morphozoic. And so this is a model that he came up with that is open source. So we have the code on our GitHub if you're interested in working with it. I think it's written in C. And so Morphozoic is a cellular automata that generates patterns that you might see in an embryo. And so we have these, it's a platform where you have not just the regular neighborhoods that you would see in a cellular automata, but extended neighborhoods. So if you're not familiar with what a cellular automata does, let me see, here it is. It's basically something like this. You have these cells that, focal cells that you want that are active. So in this case, it would be the middle cell. And this middle cell has neighbors. And it's influenced by the activity of its neighbors. So this cell here, this white one in the middle, is the focal cell. And so it's going to listen to its neighbors. But what are its neighbors exactly? So in some cases, in what they call the von Neumann neighborhood, you have these black cells, which are the neighbors, which are the direct neighbors in the cardinal directions. In a Moore neighborhood, it's every cell that touches the focal cell. So it's all of these cells around it, these 9 cells in black here. And then finally, the extended Moore neighborhood, which can go out maybe 2 or 3 layers away from the focal cell. So the idea is that these focal cells are influenced by the state, the collective state of all these neighbors. And this happens across the grid. So you have a grid where each automata or cell is influenced by its neighbors. And so you get these patterns that emerge because they're all active and they're all influencing each other in parallel. And they're basically producing these patterns across the grid. They're simple pattern formation. In some cases, like the game of life, you have things like things that move across the screen, like gliders, where you have different patterns that emerge. And so there are a lot of ways you can classify these output patterns. And so in Morphozoic, you use a neural network to classify them. There's a lot of detail to Morphozoic in under the hood in terms of what the focal cell does. There are distributions that you can analyze within the cells. And so, there's a lot of information in there. But basically the idea is that you have these sort of multi-layered networks, or these multi-layered neighborhoods that determine what the state of the focal cell is. So, and you can produce things like this, where you have this grid in there, these cells, and they're all interacting, and they form coherent patterns. And sometimes you can actually analyze these patterns as the execution of different rules. So each focal pattern will execute a rule over time based on the state of its neighbors. And so it'll be like something like, if a majority of my neighbors are off, I turn off. If a majority of my neighbors are on, I turn on. And so just with those rules being implemented, you can get patterns like this. So this is rule 30, right? So this is rule 30, where a single cell is turned on in the top row. And then the next time step, all the cells are subjected to rule 30. 2 of them change state as a result, leaving 3 cells turned on in the second row. So in this case, the rule is to have these cells executing in sequence and then producing this pattern. So there are ways you can do this that, produce these patterns. And Stephen Wolfram has a huge book on this, and he's classified, he's identified at least 200 rules that these cellular automata follow. And, they're all different. They all result in different shapes, different patterns, but they all have, we all know what the rules are and that went into creating them. So this is very analogous to pattern formation in embryos, as we've seen in some of the papers we've talked about. And the good thing about using something like Morphozoic is you can understand the rules that unfold. So if we compare that to something we're doing with image segmentation, we're trying to discover the pattern of images, right? That of a process that's already taken place. In this case, what we can do is we can actually sort of forward generate a pattern using certain rules. So we know, for example, that certain rules have certain outcomes, and if we apply them in a computational environment, we can get these, we can simulate them and get different patterns, and then understand, which patterns are maybe replicable or, aren't just ephemeral things. So this is like what you can do with CA. And so this is something that is a specialized package that we have in the group. But there are all sorts of different packages you can use for this. Everything from like a standard cellular automata model to some very highly specialized model. And again, I would recommend that you read through the paper, and it gives you a lot of detail of how this technique works. So it's a very specialized type of CA technique. We have this is what I was referring to, cell type densities. So there's in morphogen neighborhoods, you have these cell type densities that you can analyze, as well as the state of the cell and the pattern on the grid. So there's a lot of potential here. I don't think it's really been, I think it's been sort of not developed enough. I know that Tom has moved on to other things, but this is something that we might look back into. Yeah, so this is the game of life, which I was talking about before. So the game of life is where you generate these patterns, and they are mobile across the grid, and you see, you basically set up this cellular automata and you run it, and you can see these different things that look like life that are emerging on the grid, and there are different things in the game of life that you can see. You have gliders, you have other types of things that emerge, and they collide and they reproduce, and so that's another way. That's another type of cellular automata. So this is, I guess, this is a very crude introduction to this, but you can have, and this is sort of the proof of concept here using an image restoration, and so I would recommend that you read this, and then let's see if this is the one that I wanted. Okay, so this is like something where I was working on this for a project. If I can come up with the poster. This was a poster at Dynamics Days where we're modeling cellular reprogramming as a process. So the idea is these cells in culture, if you transfect them with mRNAs, they will affect gene expression, which allows them to change their cellular state. So they go from like this skin cell type to these stem cells. And so the question is how do you model that process in a way that's allows us to understand it? And so you can use cellular automata, and in this case, you're using something called sort of a sliding neighborhood, which is where the neighborhood would actually deform based on the nature of the process. So you'd have these neighborhoods where the cell would grow, and then it would influence the neighboring states would influence the cell, and it would eventually change. So there's this. There are different ways you can set up these models. That's the point of this, showing you this. And so the advantage of using a cellular automata in something like this is you can look at the spatial heterogeneity of a biological process. So in this case, you have a bunch of cells that are aligned in a culture. And when you deliver this stimulus to the cell, it will behave in a way that, different cells will take up different amounts of the stimulus. And then some of them will have different things going on inside of them that enable the reprogramming process. And so you end up with basically these colonies of transformed cells that change their state, and some cells around the edge that don't change their state or die off. And so that's the idea. We want to understand that process better. We also introduce a mathematical model to this, which is the Fitzhugh-Nagumo model, which is an excitable cell model. So you can actually model this process as a model of excitability across space. And so each cell that takes up the mRNA that you're delivering to the cell and transforms successfully is excited, and the cells that don't do it are not excited. And this is a lot of parallels with neural activity. They use this model, the Fitzhugh-Nagumo model, in modeling neural activity and neural excitability. And it's something we've been talking about to look for looking at the Basilaria colonies as well, a model of excitable media. And so in this case, it would be like, each cell, if it's moving, there's an excitation there and a relaxation. And you can model all that in a mathematical model. So this is the same one. So this is the, and then there's another poster where I think I advanced this idea a little bit more. This is where it's called dynamical cellular encodings. So this is a dynamical system where you have, again, these sliding neighborhoods, and you have these intercellular functions. So you have the focal cell and you have the neighbors, and you're annotating the cells in a neighborhood, and they're doing things across the cells. They're making comparisons. So they're not just making applying simple binary rules. They're actually applying different rules and seeing if conditions are true or false. And so you can get, you can use that type of information in a CA as well. And you can incorporate that into the design, and it'll result in different patterns. So it's not just a matter of having a grid where things turn on and off in parallel. You can apply all sorts of rules. You can even apply, within each cell, you can apply things like the Fitzhugh-Nagumo model, or you can apply some sort of gene regulatory network model, or, you can do a number of things. There are a number of ways very innovative ways you can approach cellular automata. So that's all I'm going to talk about there. I think that's, like I said, it's a very crude introduction to it, but I think it'll help people understand a little bit about, and I'll probably, if Shruti wants to do a presentation on it, she's welcome to. Otherwise, I can do a short presentation on it in the coming weeks as well. Okay, Ujjwal has to leave. Thank you, Ujjwal, for attending, and thank you, Akshay, for attending. Yes, so again, I'm going to go over a couple more things before we end our meeting. I wanted to talk a little bit maybe about some papers that I've found over the week. So last week, the Twitter thread, I put a Twitter thread for the meeting up every week with the YouTube video. And so if you're on Twitter, you want to check that out. And I have I found a thing related to cellular automata last week on Twitter, and it was this animation where using one cellular automata to feed another. And you can't see the I don't know if I have the GIF here, but basically this is where there's a cellular automata here and it's hitting a barrier. And then it's transforming the output of this automata into some other visualization. And so there's this, and I found out after seeing this, and it was very cool, but I found out that after this, there's something called cyclic cellular automata. And so these are things that recycle. So there's a sort of trophic relationship with the cellular automata and its environment. So there's this repo on cyclic cellular automata. There's this animation, and with I think with the code as well. And so if you go back to last week's Twitter thread on the meeting, there is this thing that I put together here, cellular automata as a trophic system. And I call it computophagy, which means that computational systems are eating one another or that they're being consumed. So this is the food line for cellular automata. So that's again something we can revisit. So I don't know how much I want to go into the different papers. I did want to go into the zebrafish gastrulation thing that I found as well. So this is a nice visualization of a zebrafish embryo undergoing gastrulation. And this is a zebrafish rock Twitter feed here. This is where they have the transformation from a spherical embryo in zebrafish, and you have this gastrulation stage taking shape here, where there's differentiation along this axis. And so you can see in this image, and this is the person who took these images, so credit goes to this person. And then this is the actual GIF, which I have as an MP4. So this is the process here. So you can see how it starts at the top, and then the cells work their way down around into a sphere. See like that, and it becomes more defined, and then you get this axial differentiation right along here. This is the neural crest area here. So you can see how that works. It's kind of coming together, and there's the neural crest, and there you go. So that's a nice visualization of that process. And I thought, this is what I'm trying to strive for here in the meetings, is to understand what these things look like. It's kind of hard to communicate sometimes. So I think that's all I'm going to do for today. I had a couple papers, but I think it's probably getting pretty late in the meeting. So I wanted to ask if people had any questions. Actually, let me go to the chat first. Okay, so we had Mayukh says, "Thank you, Mainak and Tharun, for the presentations. Great work. I also encourage others to present their findings in the coming weeks if possible." Yes, please do. If you want to present anything, let me know before the meeting on Slack or by email. We also have an email newsletter. I don't know how many of the new people are getting that, but let me know if you're not getting it and I can subscribe you. It just tells you about the next week's meeting. And do we have any other questions before we go?
No, I think it was good. It was a nice meeting today.
Okay. Well, thank you. And again, if you have anything you want to present next in the coming weeks, let us know. Otherwise, let's everyone have a good week. Thank you for attending. Bye.
Bye. Have a good week, everyone.